Can you please start, sir?
Okay, so how many people do I have there? Because I can only see, okay, I can see 2 participants.
Yeah, it's only myself and you. So, I mean, just go on, sir. Hopefully they will join in.
Okay.
One other person left, but she's probably going to join again.
Okay, so we're looking at classification of living things. And before we actually go into the classification proper, we need to understand a few terminologies. So there is what we call systematics, or as is written in some other sources, you can use the word taxonomy. And for taxonomy, this has to do with grouping. The key word there is grouping. It has to do with grouping of living things into their kinds based on similarities. You could also see in some places where it is said that it has to do with placing living organisms in groups called taxa. So whichever definition you choose, the whole idea behind systematics or taxonomy has to do with placing living organisms into different groups based on their similarities. So next we look at nomenclature, and nomenclature has to do with naming living organisms. In terms of the type of nomenclature we use currently, we are going to be talking about the work of Carolus Linnaeus or Carl von Linné. So that will come up later on in the lesson. But one thing I want you to take away from this particular slide is if you are being asked what systematics or taxonomy is, at least you should remember that it has to do with grouping of living organisms. And then for nomenclature, it has to do with naming. So the keyword for nomenclature is naming. Next, we look at Carl von Linné, who was a Swedish naturalist that lived between 1707 and 1778. So it was that proposed this 8-stage classification system, which we use till today, although this has undergone quite a number of modifications. But at least the groundwork was laid by Carl von Linné. You may not particularly see the name Carl von Linné. You may actually see the Latinized name, which is called Carolus Linnaeus. And so this is called the Linnaean classification system. So what does it involve? Like we said earlier, we're talking about systematics being grouping living organisms into different groups, all right? And that is based on their similarities. So in order to achieve this, we are going to put these organisms into this 8-stage classification system. This means that if we're talking about kingdom would have the largest number of members. And if we're talking about species would have the smallest number of members. And then if we are talking about the level of similarity, if you say 2 organisms belong to the same species, for instance, it means that they are most closely related. Okay, so the least related will be those ones that are in the same kingdom. So next, we move on to the binomial nomenclature. Now, if you look at this slide very well, you will see that there are 2 things that are credited to Carl von Linné. One is developing this 8-stage classification system, and secondly, the development of the binomial nomenclature. So the word bi simply means 2. So when we talk about the binomial nomenclature, we are talking about a type of nomenclature that assigns two names to living organisms. So which names are we talking about? There's what we call the generic or the genus name, and then there's what we call the specific or the species name. So for instance, if I say Homo sapiens, the word Homo would be the genus name, and the word sapiens would be the species name. Take note that when you write these two things, they have to be underlined or italicized. Since I cannot properly write in italics, I prefer to underline them. And then take note also that the genus name starts with a capital letter, while the species name starts with a small letter. All right. So that's an example of the application of the binomial nomenclature. Let's look at the work of R.H. Whittaker. R.H. Whittaker lived between 1920 and 1980, and he came up with these 5 kingdoms that we talk about today. All living organisms will fall into any of these 5 kingdoms. So we have the kingdom Monera, which has to do with the prokaryotes. The prokaryotes are your bacteria and the blue-green algae. We also talk about Protista. Protista includes organisms like the protozoans. Next, we talk about fungi, which includes organisms like mucor. And then finally, okay, there's still another kingdom that is missing, which is kingdom Animalia. So for kingdom Plantae, we're talking about the plants and bryophytes as well as pteridophytes, which we'll talk about later on. For kingdom Animalia, we're talking about animals like your fishes, which we consider under the umbrella group Pisces, the amphibians, the reptiles, the mammals, etc. So again, to recap, the 5 kingdoms that we talk about today was developed by R.H. Whittaker, and all living organisms must fall into any of these 5 kingdoms. So to look at each one more closely, we'll start with Kingdom Monera. Now, the Kingdom Monera are the ones we refer to as the prokaryotes. And when we see the word prokaryotes, what we are, in fact, the first thing that should come to our minds is the fact that they do not have a nucleus. So out of all the 5 kingdoms, it is only Kingdom Monera that we can refer to as prokaryotes. And that simply means that they do not have a nucleus. Of course, they have other notable characteristics. One, they are unicellular. They could be motile or non-motile. To clarify the meaning of the word motile, okay, motile means actually mobile. We do not particularly use the word mobile for unicellular organisms. Rather, we use the word motile. So we don't say that unicellular organisms are mobile. We say that they are motile. Again, like I said earlier, unicellular organisms that are associated with the kingdom Monera can either be motile or non-motile. Okay, so we look at other interesting features of the kingdom Monera. They lack membrane-bound organelles. And when we say that they lack membrane-bound organelles, what we really mean is that they do not have organelles like mitochondria. They do not have organelles like lysosomes. They don't have organelles like, let's say, endoplasmic reticulum. So any organelle that has a membrane is lacking in a typical prokaryote. Of course, the fact that they lack membrane-bound organelles does not mean that they don't have some other unique features, okay? For instance, they have what we refer to as plasmids, and plasmids are another form of DNA that has genes that confer certain unique characteristics onto this prokaryote. Okay, some of them have flagella, some have cilia. Generally, they all have a cell wall. Okay, and your typical examples of the kingdom Monera would be things like maybe E. coli, Staphylococcus, Streptobacillus, blue-green algae, like the nostoc and the oscillatoria. So what can we take away from this slide? One, when we talk about kingdom Monera, we are talking about the only kingdom that has members that do not have a nucleus. Secondly, we are talking about a group of unicellular organisms that do not have membrane-bound organelles. Now, yes, they have DNA. Some of them may have flagella or cilia, and yes, they also have cell wall. Moving on, we talk about the kingdom Protista. And for the kingdom Protista, like I said earlier, we are talking about the protozoans. Now, one unique thing about this kingdom Protista is we have groups that have animal-like properties, and we also have other groups that have plant-like properties. So, for instance, when we talk about examples like Euglena and Chlamydomonas, these protozoans tend to have plant-like properties, like the presence of chlorophyll, which could be found in the organelle chloroplast. Of course, we also have other ones that have animal-like properties, like amoeba and paramecium. So generally, what should we remember when we are talking about kingdom Protista? We already know that they are not prokaryotes, which means that they have a defined nucleus. By the way, if you're confused about the meaning of the word nucleus, nucleus simply means that there is a membrane around your DNA. So for kingdom Protista, there's a membrane around the DNA. Now, secondly, since they are eukaryotes, of course, they would have membrane-bound organelles. And we can see this even in the diagram. You can see that they have Golgi bodies, they have chloroplast, they have mitochondrion. All these are membrane-bound organelles, which shows that the members of the So which shows that the members of the kingdom Protista are a bit more advanced than the members of the kingdom Monera. All right. So 1, they are eukaryotic. 2, they have a nucleus. 3, they have membrane-bound organelles. 4, they are single celled. They are only made up of one cell. So we can refer to them as unicellular. And then 5, we do have some of them that have plant-like properties and others that have animal-like properties. In terms of the kingdom fungi, we are talking about things like rhizopus, muco, mushrooms, aspergillus. All these are notable examples of the kingdom fungi. So what are the things we need to take away from this particular kingdom? Unlike Monera and Protista, kingdom fungi has both unicellular and multicellular forms. Now, the unicellular forms that obviously you would be familiar with would be things like yeast. And if you were in doubt before now, yes, yeast is actually a fungus, not bacteria. So we start by establishing the fact that the first property we're going to look at, or the first feature, is the fact that the kingdom fungi could be unicellular or multicellular. Next, they equally have a cell wall that is made up of chitin. Now, chitin is not cellulose, so we should not confuse the two, okay? The cell wall that we find in the fungi is called chitin, or is made up of chitin. For fungi, they reproduce sexually and asexually, while some of them reproduce sexually by conjugation, okay? Many of them reproduce asexually by spore formation. And again, this brings us to another issue. What exactly does the word spore mean? Now, if you look at the diagram here, you can see these circular structures that are being released from this large spherical structure. Now, these small spherical structures that you're seeing here, we refer to them as resting forms. And the beauty of being able to produce spores is that when they land on wherever they land, if the conditions are not to their liking, they do not germinate. So they remain in that resting or dormant form until they find conditions to their liking, after which they begin to germinate. So we should take away from this particular slide that one way that the kingdom or the members of the kingdom fungi reproduce asexually is by the production of spores. Next, we consider the fact that members of the kingdom fungi feed by saprotrophic mode of nutrition. What exactly does the word saprotrophic mode of nutrition, what does it mean? It simply means that if this particular fungus, for instance, is going to feed, it would release enzymes onto the substrate. These enzymes will digest the complex molecules in that substrate and then absorb the simple soluble forms that have been produced as a result of that digestion process. So that is what we call saprotrophic mode of nutrition. Before we go on, there are two things we need to equally look at when we appreciate the structure of the fungus. Looking here, we can see that there are stalk-like or thread-like structures, okay? these things, in actual fact, when you look at them under the microscope, they actually occur like threads. So we say that they are thread-like. And these thread-like structures, as you can see, we refer to them as sporangiophore. Okay? Now, at the terminal end of this sporangiophore, we have sporangium. Within the sporangium, we have the spores. So when the sporangium is ripe, it would release the spores, and the spores will now be dispersed by wind. But beyond all the structures that I've also itemized, we also need to be able to label this structure, which is called the stolon. It is called a stolon because it is more like a stem that is creeping above the surface of the substrate. Now, the part that you see within, embedded within the substrate, is what we refer to as the mycelium. Okay? And then the part that is above the substrate, we refer to that as the hyphae. So all this is a hyphae. This is another hyphae here. So these are certain things we need to take into consideration when we are trying to remember one or two things concerning fungi. Of course, like I said earlier, the examples of the kingdom fungi include mushroom, rhizopus, and mucor. So what are the things we need to remember as far as this kingdom is concerned? They could be multicellular or unicellular. In terms of structure, they appear as thread-like structures, which we call hyphae. They can reproduce sexually or asexually. Most importantly, when they reproduce asexually, they produce spores. And then finally, their mode of feeding is what we call the saprotrophic mode of feeding, which involves extracellular digestion. Now, let's try and answer one or two questions on this particular topic. Now, of course, your knowledge of the previous topic is also going to come to bear as far as this is concerned. But if there is need be, I would go back and refresh your memory. So can you answer this question? Euglena may be classified as a plant because it, first of all, Euglena belongs to the kingdom Protista. And I said, when I was talking about the kingdom Protista, you have forms that have plant-like characteristics and members that have animal-like characteristics, okay? Euglena is unique in its own case because it has both plant-like and animal-like characteristics. So if you were going to supply an answer to this question, what would your answer be? Amina. Amina. Can you hear me?
Yes.
Yeah, so what would your answer be to this particular question?
Sorry, I joined late, so I did not join the rest of the, from the beginning. I joined late.
Yeah, but you were...
But I can guess because
yeah.
it has plant characteristics.
Mm-hmm. So what would your answer be? Hello?
Excuse me, sir. I will
Yes.
say C. I don't know. Do just a guess. So, I mean, I
Okay.
just wanted to
So,
tell the students not to be shy. It's fine if you don't
okay.
get it right.
Yes.
Yeah. Okay. We're all here to learn.
Yeah. So, this is what euglena looks like. Okay. And I'm seeing your name there. Okay. So you may see some things like this. These things are called chloroplast. This is the flagellum. Okay. Now, the flagellum is actually an animal characteristic. Okay. One plant characteristic that you cannot argue with is the presence of chloroplast. All right. So obviously, when we say that euglena may be classified as a plant, it simply means that it has chloroplast. Every other one that you look at here, presence of a vacuole. Vacuoles are associated with animal cells. Pellicule provides a definite shape. Okay, but pellicule is something that is also associated with animal features. Flagellum is associated with animal features. So the one you cannot really argue with is the presence of chloroplast. Number two, which of the following is the lowest category of classification? Amina, over to you.
The least, the
Yes.
species, right?
Yes, that is the species. Yeah. So this, you got this correctly.
Yeah.
Yeah. So moving on, let's look at the kingdom Plantae. We've looked at two kingdoms already. We've looked at kingdom Monera. We've looked at kingdom Protista. Okay, so we are moving on to kingdom Plantae. All right. And in kingdom Plantae, one thing that generally cuts across all members of the kingdom Plantae is the fact that they are multicellular, they have chlorophyll, and they have a cellulose cell wall. Okay, so this is what all of them have in common. But be that as it may, we still have some groupings associated with this particular kingdom, all right? We have what we call the bryophytes, okay? For the bryophytes, we have what we call the mosses and liverworts. These are the typical example of your bryophytes. We have mosses and we have the liverworts, okay? And then we have the seedless plants. They are vascular, but they are seedless. And here we talk about things like the fern. Then finally, we talk about the vascular plants, all right, which are seed bearing. I'm still going to clarify these two groups as time goes on. But for now, at least we should have at the back of our minds that when we talk about the kingdom Plantae, we're talking about bryophytes and we are talking about tracheophytes. But before we even go on to bryophytes, there is another group that we refer to as the thallophytes. So one, talophytes, 2 bryophytes, and then 3 tracheophytes. All right. Now, we're not doing any revision question yet. So let's have a recap of what I just said. I said we have 3 groups under kingdom Plantae. We have the talophytes, the bryophytes, and the tracheophytes. So let's start with the simplest of all of them, which is the talophyte. And when you hear the word thalo, what should come to your mind is the word thallus. So when we say thallus, we are saying that we have flat bodies like this one here, or filamentous, bodies like what we have in Spirogyra. Okay, so they could have flat or filamentous bodies. Those are the kind of organisms or plants that we find in the division Thallophyta. Okay, of course, because they are plants, all of them have chlorophyll. But also, because they are not advanced plants, they don't have roots, stems, or leaves. Okay? Like I said, because they are not advanced plants, they don't have root, stems, or leaves. Also, because they are also not advanced plants, they lack what we call vascular bundles. Okay? And what do we mean by vascular bundles? Vascular bundles, we talk about the xylem, the cambium, and the phloem. Okay, xylem transports water and minerals. The phloem transports food like sucrose and amino acids, while your cambium is what we call a meristem that divides to produce the xylem and the phloem. So your typical examples are what we call the red algae, the brown algae, and the green algae. Okay, of course, if you don't want to talk about that, you can just pick on any other example that you are seeing in front of you here. But like I always do, what can we take away from this slide? When we talk about the division Thallophyta, we're talking about plants that have flat or filamentous bodies. We're talking about plants that are not advanced. Not advanced in what terms? Not advanced in the sense that they do not have roots, stems, or leaves, okay? And not advanced in terms of the fact that they do not have vascular bundles. So those are the things we need to take away from this particular slide. Then we look at other examples of the members of the division Thallophyta. You may be familiar with Chlamydomonas. Sorry about this spelling. Chlamydomonas is C-H-L-A. Okay, so that is Chlamydomonas. You can be familiar with Volvox, Eulothrix, Ulva. So all these are also examples of the division Thallophyta. So let's move on to something a little bit more advanced, which is the division Bryophyta. And the common examples of this particular division are the mosses and the liverworts. Okay, if you can remember hornworts, oh well and good. So what do we need to know about the division Bryophyta? One, they grow in very damp places. Okay, and then 2, Their bodies are made up of rhizoids and a shoot-like structure. Now, the rhizoids, you cannot particularly see them here, but I will show you what the rhizoids look like in the next slide. And then 3, they lack true roots, stems, and leaves. Now, take note that we said they lack true. What this means is that they actually have structures that look like roots. They have structures that look like stems, and they have structures that look like leaves, but they are not really as they appear. Those ones that look like roots are not roots. Those structures that look like stems are not stems, and the ones that look like leaves are not leaves. And that is why we say that for bryophytes, they do not have true roots, stems, and leaves. And then one notable aspect of this bryophyte is the fact that they exhibit alternation of generation. Now, when we talk about alternation of generation, we're talking about alternating between what we call a sporophyte phase and a gametophyte phase. This I would explain in the following slides. So this is a typical bryophyte here. In fact, this is a moss, right? This is a moss plant. And when you look at this moss plant, one thing that strikes you immediately is the fact that it actually has 2 major sections. Okay, it has what we call a sporophyte, and then it has what we call a gametophyte. Now, if you look at this diagram very well, and let's say you were looking at this thing in real life, which part do you think will stand out more? Amina?
The sporophyte?
As in, if you were looking at it in real life, which part do you think is more would be most conspicuous? Would be more visible to you?
Part A? Part B. Oh. Part B.
Exactly. So this is the part that, because this part you will not really see. You know why you're able to see this very clearly? Is because it's a diagram. But in real life, you would not be able to see this as clearly as, you know, it is represented here. So because the gametophyte is the part that stands out, we say this is the dominant phase or the dominant generation. Okay. Now, of course, we have other aspects that you need to take into consideration. The fact that you have this stalk-like structure, which we refer to as the seta, and then we also have this structure, which we refer to as a capsule. The capsule is that structure that contains the spores. And yes, if you have a sporophyte, it definitely means that you are going to reproduce by producing spores. Okay. So here we have what I described as the alternation of generation. And of course, you need to have an idea of reproduction in bryophytes, okay? We already know that this is the, sporophyte phase, and we know that this is the gametophyte phase. Now, this thing that you have here that we call rhizoids, they look like roots, but they are not roots, okay? Now, within the capsule, like I said earlier, you are going to have the production of spores, okay? So when the capsule is ripe, it falls off and releases the spores, okay? Now, these spores, like I said, when they fall in an environment or onto a substrate where conditions are to their liking, they will germinate into what we call a protonema, okay? And this protonema will develop into a gametophyte that will have both male and female sex organs. Let me remove this. Okay, so it will have both male and female sex organs. Now, this archegonium here is the female sex organ, whereas the antheridium, which is not the antheridium that you have here, is the male sex organ. So the sperm cells, what we call the antherozoid, will travel from the antheridium and will travel all the way into the archegonium, where it is going to fertilize the egg. And once the egg is fertilized, it develops into a zygote. Now, one very important point here is the fact that bryophytes, due to their nature, are heavily dependent on the availability of water. Why? Because the sperm cells have to swim to fertilize the egg within the archegonium. So that is why they require a very damp or moist or watery environment. In any case, when the zygote is being formed, it will develop into a new thallus. Okay, and this new plant, as usual, would have a gametophyte and a sporophyte phase. So this gives you an idea of what we mean when we talk about alternation of generation. So let's look at the division Tracheophyta. Again, we've looked at kingdom Monera, we've looked at kingdom Protista, we've looked at Bryophyta and Talophyta under kingdom Plantae. Next, we are going to look at the division Tracheophyta. And again, when you hear the word tracheo, as if to say trachea, okay, what should immediately come to your mind is vascular bundles. Okay, that is what you immediately come to your mind, vascular bundles. All the members of the kingdom, sorry, the division Tracheophyta possess vascular bundles. Now, you may be wondering, I've been mentioning the word division. That is because in plants, after kingdom, we don't particularly use phylum. Okay? For Americans use the word phylum for plants, but in Nigeria, we don't use phylum, we use division for plants. So in any case, let's look at the tracheophytes. So these can be subdivided into the seedless plants and the seed-bearing plants. So which ones are the seedless plants? The members of the subdivision Pteridophyta. Which ones are the seed-bearing plants? The member of the subdivision Spermatophyta. Okay, so we're going to look at each of them in detail. I am very sure you are familiar with division Pteridophyta. I'm sure you've seen these plants around you because it's a very, very common plant. So let us try to unpack the little details associated with this plant. One, it is terrestrial. Now, why are we mentioning the fact that it is terrestrial? This is very important because when we talked about bryophytes, even when we talked about talophytes, there is a dependence on water. But at this particular point, right, you find out that we are emphasizing the fact that the pteridophytes are terrestrial. So they are not really dependent on, of course, to an extent they are, but not as dependent as the bryophytes and the thallophytes. In any case, we have a true root, a true stem, and true leaves, unlike what we had in bryophytes. Okay, for bryophytes, we did not have a true root, we did not have a true stem, we did not have true leaves. For pteridophytes, we have true roots, stems, and leaves. Now, then they have a rhizome. Before I go on, I need to distinguish between the word stolon and the word rhizome, okay? So let's say this is a particular surface here. If you have an underground stem, we call this a rhizome. If your stem is creeping above the surface, we call this a stolon. So if you look at the fact that your pteridophytes have underground stems, that is why we use the term rhizome. So they have rhizomes under the ground surface, okay? And then from these rhizomes, we have certain things that emanate at certain points of that underground stem. These we refer to as fronds. In any case, for us to fully appreciate the structure of the pteridophyte, we need to go to the next slide, okay? So this is a typical pteridophyte or fern. We have an underground stem, so let me draw a line. Let me draw a line that will show you the ground surface, okay? So you can see that we have an underground stem, that is the rhizome. And then emanating from this underground stem, right, we have the frond. As you can see, the frond includes the stalk and the blade, okay? Now, the blade is that leafy aspect of the frond. Zooming into the blade, we will see what we can refer to as the pinna, okay? So the pinna is what we can describe as the leaflet, the small leaves that we find here, all right? Within each of these leaflets, we have what we call the soros. Within the soros, of course, the plural for soros is sori, all right? So within each soros, we have the sporangia. Okay, sporangia is plural for sporangium. So the singular is sporangium. Now, within each sporangium, we have spores. So looking at this again, if we say, let me even ask you, can you describe the structure of a fern? Amina.
Yeah, I sent message just now. I can't see the slide. It's still
You can't see the slide. What are you seeing in front of you?
Yes, I can't see the ones you're explaining right now. The recent ones.
Okay, so what are you seeing in front of you right now?
front of me is the slide about the life cycle of the bryophyta, something like that. Okay, I can see. Yeah, fixed.
Okay, what do you see right now? Okay, you can see it now.
The pin, something like that.
No,
Yes.
can you tell me some of the words you are seeing?
Sporangia, sorus, frond, stalk, rhizome. Is that it?
Okay, what about this? Were you able to see this?
Yes, I'm able to see it.
Okay. Please, sir.
Yeah?
I think I could also not see the slides that you are explaining. Anyway, we can fix that later on.
Okay.
Let's carry on. We'll finish in about 7 minutes time. Thank you very much.
Okay. Okay. So what I was talking about is the structure of a fern, an example of pteridophyte. You know, we've talked about the fact that for plants, right? I started by talking about the thallophytes. From there, I moved on to the bryophytes. From the bryophytes, I moved on to the tracheophytes. And under the tracheophytes, we have the pteridophytes, which are the seedless plants. And then we have the seed-bearing plants, okay, which are what we refer to as the spermatophytes, right? Now, for the pteridophyte, like I said, they have an underground stem, and that is why we use the term rhizome. Now, from this stem, you can see what is emanating. There's a structure emanating from this stem, which we refer to as the frond. Okay. Now, the frond consists of the blade and the stalk. Okay. Now, the blade, as you can see, is like the leafy aspect of the frond. When we zoom into the blade, we will see that it consists of leaflets, right? Now, each of these leaflets consists of what we call a sorus. And the word sorus is singular. The word sori is plural. So each sorus consists of sporangia. Like I said, sporangia is plural, sporangium is singular. Within each sporangium, we have spores. Okay, so these are little things that you need to take into consideration when you are trying to remember what a fern looks like. Okay, so I'm going to round up with this. What reproduction in your fern or pteridophyte. Now, this may look like reproduction in bryophyte, but there are some slight differences here. Okay, there are some slight differences here. All right. Now, fine. Here we have the production of spores as well. That is a similarity between what we have in bryophyte and what we have in pteridophyte. Okay, and yes, the spores are coming out of a sporangium. That is also a similarity. But when we consider the fact that in your bryophyte, the spores were coming from the capsule, okay, and that capsule was being carried by what we refer to as a setter. If we look at the pteridophyte, you find out that each of these leaflets, okay, is containing a sorus. And the spores are actually coming from the sporangium in the sorus. In any case, once it is time, spores are released. the spores develop into this heart-shaped structure. You can see that it looks like a heart. Okay. So this heart-shaped structure is what we call the gametophyte. Okay. Now, this gametophyte contains, as usual, the antheridium, which is the male part, and the archegonium, which is the female part. And as usual, the sperm cells arise from the antheridium, they migrate to the archegonium, where they fertilize the egg to produce a zygote. Okay. Once this happens, you find out that this gametophyte now begins to develop a sporophyte. Okay. And eventually also develops this structure we refer to as an underground stem. Okay. Now, if we look at this is where I'm going to round up for today. If we look at this, okay, we can easily see that the part that stands out, okay, is actually the sporophyte as opposed to the gametophyte. Now, the sporophyte being the part that contains the spores, okay? That is the part that stands out conspicuously as far as ferns are concerned. So the dominant generation, as far as ferns are concerned, would be the sporophyte. Okay? As opposed to what obtains in the bryophyte, where the dominant generation is the gametophyte. So we'll round up with this question. Amina, are you there?
Yes, I'm here.
Yeah, so substances that conduct materials throughout a plant are called?
The vascular bundles.
Yeah, pretty much. That is it. Okay. What about number 2?
The answer is already highlighted. It's A. Is it?
It's not A.
In
You know what is?
Okay, let me read the
sex
question.
organs.
Let me read the question. In birth type, sex organs are produced in the rhizoid, I guess.
No, the rhizos is the part that looks like the roots.
Okay, it's definitely B because the gametes, right?
Yeah, so the gamete, yes, it's the gametophyte. Okay, that contains, yes, it's the gametophyte. Can you remember the male part of the gametophyte? Can you remember the male part?
Yeah.
What's the name? Antheridium.
Not, is it? The names are.
Yeah, the male part is the antheridium. The female part is the archaea.
The names are quite hard to remember, but I think it starts with A and something like that. Yes.
Okay.
Yeah.
So the male part is
antheridium. Yes.
Yeah, and the female part is archegonium.
Yes.
Okay.
Okay.
Yeah, so that's where we are going to round up for today.
Okay.
Yeah.